Okay. So this review, this is going to be over chapters 5, 6, 8, and 9. 5 is over the eukaryotes. 6 is over the viruses. 8 is metabolism, and 9 is going to be genetics. We have this just like the last one with some questions associated with each of those. All right, let's jump into the eukaryotes. Our first question is, what is the defining feature of eukaryotes? So this is definitely asking us to compare eukaryotes to prokaryotes, and the difference, of course, is that eukaryotes have a true nucleus, so it's a membrane-bound nucleus, and that's a big difference. They also have membrane-bound organelles. That counts. That moves us on to explain the endosymbiotic theory. With the endosymbiotic theory, the idea is that there was an old precursor cell that ate up like something that was like a bacterium, and they had a symbiotic relationship, and then things have stayed that way forever. And that has led to the development of mitochondria and chloroplasts, if not other organelles. So that's the idea with the endosymbiotic theory, and the reason that they think it, works is that mitochondria and chloroplasts have their own chromosomes that are circular, just like a bacterial chromosome would be. Single circular chromosome. They have to divide themselves. They're not going to have, the cell won't divide them. So they have to do that on their own whenever the cell is replicating. There's other stuff too. I'm trying to remember it off the top of my head, of course. So the ribosomes that are present inside of mitochondria and chloroplasts, they have their own ribosomes, and they're like bacterial ribosomes and not like eukaryotic ribosomes. So those are some things that are evidence for the idea that our mitochondria, which are energy powerhouses for the eukaryotes, that those came from a bacterium that was able to produce energy for us, and the chloroplasts, which are going to be involved in photosynthesis for eukaryotes, that those came from some sort of photosynthetic bacterium. It was beneficial enough that that became a permanent relationship. So our next question is, true or false, bacterial flagella are pretty much the same as eukaryotic flagella, and we just talked about that today in our motility lab. Remember, bacteria flagella are going to be rotating in like a motor. They'll go clockwise or counterclockwise, and they'll determine the direction that they can be moving, whereas eukaryotic flagella are much larger, and they can move in a whip-like fashion, not rotating. And they have microtubules, which are essentially structural filaments for the eukaryotic cells. Those microtubules are going to be arranged inside of the flagella to support it and give it structure. So cilia are like little flagella, essentially, little eukaryotic flagella. They're tiny little ones, and you'll have like usually a whole lot of them. They're sometimes associated with locomotion. This is just for the eukaryotes. They're sometimes associated with locomotion, but they're more commonly associated with like feeding and like filter feeding. So filtering stuff out of the whatever water that they might be living in, for example, and to get nutrients out of it and catch it in the little cilia for absorption. Okay, the glycocalyx in prokaryotes, in bacteria and archaea. The glycocalyx is what gives us the capsule and something called the slime layer, which is like a loosely associated capsule, essentially. So it's like sticky kind of coating. It's what it sounds like. It looks like a slime layer. So we don't necessarily have that quite the same way in eukaryotes. Some eukaryotes can have a capsule. When we're talking about multicellular organisms like us, we do still have glycocalyx, but it's usually associated with our extracellular matrix, which helps keep the cells, associated with one another and helps define organs as well. What do eukaryotes? Sorry, I need to probably mark these off as I go. Do eukaryotes have cell walls? Yeah, some of them do. Like plants have cell walls. Those are made of cellulose. We also can have cell walls in the fungi. Fungi can have a chitin cell wall or cellulose cell wall. So those guys can have cell walls too. So they're not the same as the prokaryotes. They do not have peptidoglycan. Molecules that provide rigidity to eukaryotic cell membranes. This is talking about the sterols, and we would point, primarily at something like cholesterol. A lot of the other sterols are going to be involved in as structure for like hormones. I really need to mark it off. I said it once. All right, the nucleolus. The nucleolus is not found in the prokaryotes. The eukaryotes have the nucleolus. And so this is going to be the region inside of the actual nucleus where ribosomal RNA is being manufactured. And it's done typically in a tight little region where you can actually stain it and see it within the nucleus. So that's what the nucleolus is. With prokaryotes, they don't have a nucleus, they don't have a true nucleus. But the region in which they keep their genetic material, they call the nucleoid region. So don't confuse that with nucleolus or nucleus. Those are three separate things. True or false? Mitosis is exactly like bacterial binary fission. No. So bacterial binary fission, they're just making a copy of their chromosome and then pinching the cell in half. That's it. And they have copies of the cells. We have to worry about our organelles. We have to worry about all these different chromosomes and making sure that each chromosome has been replicated and doubled from a single to, that double copy, so that when we pull it apart in mitosis, it'll actually, have a copy in each side. All of that stuff has to be regulated in a much different way than binary fission would be dealing with that. So the steps of mitosis, I remember this as PMAT, like dog has to go pee on the mat instead of on the carpet. So this is prophase, metaphase, anaphase, and telophase. And those are basically in prophase, we're going to be having our chromosomes replicated and getting everything ready for the whole process. The metaphase, we're lining up our chromosomes in the middle and attaching the spindles and the centrioles to get ready to pull them apart. Pulling apart happens in anaphase. And then telophase, we're going to just start consolidating everything back into two separate cells, reforming the nucleus and pinching off the cells separate from one another. The checkpoints in the cell cycle, there's all these different checkpoints just to literally make sure that the genome, the DNA that was made when we were copying it or before we copy it, making sure that there are no mutations in there that shouldn't be there, so that we can go ahead and repair those before we make new daughter cells and spread that potential mutation on. The difference between rough and smooth endoplasmic reticulum, the rough endoplasmic reticulum has ribosomes all over it, and that part is going to be the part that's involved in protein production. The smooth is going to be involved in producing other molecules, non-protein molecules. The Golgi apparatus is where you go after you are made in the rough endoplasmic reticulum. Your little proteins are going to go to Golgi apparatus for modification, as well as packaging and transport within or out of the cell. A lysosome and a vacuole. A vacuole is more or less a large storage compartment within the eukaryotic cell. It's often where plant cells store their water. And a lysosome, so if you have phagocytosis, which is large, engulfing of something by the cell, and you take it up by phagocytosis and you make it into a little vesicle. Now, whatever it is, your pathogen that you ate up or whatever, broken up cell components from the nearby dead cell, whatever you ate up, you take it into this little vesicle called the phagosome. So that's all that's called until it fuses with a lysosome. So the lysosome contains all of those digestive and toxic things that will kill the bad guys, the pathogens, or break down, cell material or something like that. So that's the idea of the lysosome. So it can have potentially toxic, damaging products, as well as something called lysozyme, which specifically targets peptidoglycan. All right, mitochondria, that's going to give us our energy, which we'll talk about in chapter 8 once we get to that here in a moment. And what evolutionary traits does it possess? Basically, that's just referring back to the endosymbiotic theory, which we already went over, so we don't need to retouch on that. True or false, if you have chloroplasts, you don't need mitochondria? Well, we know that chloroplasts allow us to harness the energy from the sun and activate certain molecules that will get excited and allow for production of some energy. Well, it only makes two, in the typical process, it only makes like 2 ATPs. That's our output from that situation. not very much energy, and it's just enough energy to make something like organic molecule from an inorganic source, like carbon dioxide. So they take carbon dioxide, they use energy from the light process in order to fuel making the actual glucose. And so they make glucose. So if you guys don't know where glucose goes after this, then I don't know what to tell you at this point, because that's what we get into with the actual metabolism chapter. So that is where the glucose is going to go in plants as well. So into aerobic respiration. So yeah, you do need mitochondria. That's what'll do that for the plants. The chloroplast is just going to generate that glucose that we'll put into the mitochondria. What kingdom does yeast belong to? Fungi. So yeasts are fungi. Don't forget that one. Hyphae and mycelium. What's the difference and what are they? The hyphae, these are thread-like structures that make up body of the fungi. So it's the part that actually makes up the body, whereas the mycelium is referring to the body, the whole total body. So that's the difference. The hyphae are sort of parts of the mycelium. The possible nutritional sources for fungi, we have saprobes, which are going to be breaking up, dead things. We have the heterotrophs that are going to be parasitic. So those could be the ones that are going to be feeding on, like athlete's foot or ringworm or something like that, right? Those are parasitic fungi. Then we have the non-parasitic heterotrophs. So that's going to be ones that don't break up saprobes, but they're not parasitic either. So they just be living off of other resources in the environment. Sporangiospores and conidiospores. Basically, what I want you guys to remember about these, they are, first of all, asexual reproduction mechanisms for fungi. Asexual. And the spores that they are making in these situations are nothing like endospores. These are like actual, reproductive situations. So we're already like, active and doing things. Whereas remember, endospores are basically dormant. That's a completely different scenario. In fungi, the spores that they make here are for reproduction, for asexual reproduction. There's two structures you would want to be familiar with here, the sporangiospores and the conidiospores. Sporangiospores have like a coating on the outside covering like the spore apparatus, whereas the conidiospores have it almost look like a dandelion. So there's out, but they're not covered. So the spores can just go freely at any given time. So those are the different structures there. Algae-based plankton are responsible for the majority of the Earth's oxygen. That is true. It's going to be the algae-based plankton that do that. They make most of our atmospheric oxygen, about 70%. Are algae fungi or are they protista? Algae are actually protista. So that's another one. That's like the catch-all kind of grouping. That includes, protista includes the algae as well as the protozoans. So that's one to remember as well. Bacteria sporulation is pretty similar. So this is the making of endospores. So those are those structures for bacteria that provide that sort of hardy protective structure, protects the genetic material in harsh conditions. Similar to that, protozoa have a structure that they call cysts. And that's almost the same thing as an endospore, functionally speaking, very similar to an endospore. When we talk about sporulation and endospores, the actual metabolically active version of those cells are going to be called vegetative cells. And those vegetative cells, they're typical what we would think about bacteria, alive and doing stuff. For protozoa, the vegetative equates into what we call trophozoites. Troph has to do with eating. So just think of those as being the ones that would be eating. The cysts aren't going to be eating. Trophozoites will. So they'll be the active ones. Which group of protozoa are non-motile and are parasitic? This is going to be the sporozoa. That's just the name of them. So there you go. And then that's going to include Plasmodium, which is cause for malaria. What are the two types of helminths and their phylum names? And then breaking it down even more, basically. So we have the roundworms and we have the flatworms. The roundworms are called the nematodes. The flatworms are called platyhelminthes. I always just remember it because platy sounds like flat to me, but so that's the flatworms. And then we can divide up platyhelminthes into another two types, which are the tapeworms and the flukes. So those are going to be the cestodes or the tape toads, and the trematodes are the flukes. So let's go on to chapter 6. I don't know where my cursor went there. All right. This is going to be talking about the viruses. You can see we have quite a few, not quite as many questions for this one. That's kind of a good thing. All right. So what is meant by filterable virus? Sort of how viruses were discovered, and you know, we kept filtering things, but then, you know, it still led to infection or something like that. Infectious agents were still there after we filtered. That is what we mean by filterable virus. It filtered out the bacteria. The virus went through, but it was clear. It should have been sterile. You know, and what we would call, you know, it was filterable. It was filtered. It looked clear. It's sort of like when I was in pitch black. It looks clear. So, anyways, obligate intracellular parasite. That means that it is obligated, so it has to do the thing. Intracellular, inside of the cell, and it's a parasite. So it's going to cause harm to the host that it's living within. So that is what all viruses are. All viruses are obligate intracellular parasites. No virus can exist or reproduce outside of the host. And specifically even can't reproduce without being inside of the host cell. So it needs all the parts of the host cell in order to basically hijack it and make the viral particles. All right, the basic parts of a virus. All you need, minimum bare bones for a virus, is the nucleic acid, whatever that is. So it's the genome. So the nucleic acid, as well as the capsid, and that is a protein coating that covers the nucleic acid. So those are the only two parts you absolutely have to have. You usually have spikes. Those are glycoproteins, so sugar plus protein, that act as keys, essentially, to get into their target host. And those keys can be extremely specific, or they can allow them to get into a lot of different kinds of cells. It just depends on the virus. So those are the viral spikes. So that's why we're talking about that. Some viruses have an envelope that they steal from the host cell when they're budding out and creating new viruses. They just push through and pinch off, coating themselves in the envelope from the host cell. That envelope has been studied with viral proteins. There's no more host proteins on there. So now we've coated our little virus particle with an envelope, a membrane from a host, and that is going to be studied with viral proteins, including the spikes that would be letting you get into the cell. So if you have an envelope, your spikes have to actually be put on the surface of your envelope. Otherwise, it's being covered. So can't do that. If you only have a capsid and a nucleic acid, then that's called a naked virus. There's no envelope, basically. And then that combination, so even if you are an enveloped virus, you still have a capsid in there that is still coating your nucleic acid. And that combination of nucleic acid plus the capsid, that protein covering, that's called a nucleocapsid. Those are just terms associated with this. So viral genomes, what do viral genomes look like? They can be RNA or they can be DNA. They can be single-stranded or they can be double-stranded. And the orientation of the single strand, if we have single strand, can be positive, so it can be read like a messenger RNA, or it can be negative, and where you would have to make a copy of it in order to read it first. So there's all these different ways that you can have all this different stuff for a virus. But viruses have either RNA or DNA, not both. Major steps of infection include adsorption. Adsorption is this basically docking with the host cell. So the cell you're about to infect, that's the docking part, making sure your key goes into the lock. And we have penetration that can be often by endocytosis. So whenever you're docked to the cell start engulfing you, essentially. And then we have uncoating, which is the cell is going to start, as it sees you as foreign or something that it just ate, it's going to start dissolving you. But that's removing your envelope and your capsid. So now your genome can start doing the work inside of the cell. So that's the uncoating portion. The synthesis portion is talking about making copies of the viral genome, as well as generating the proteins needed to make new virus particles. And pretty much the entire cell is going to be dedicated to these things and nothing else, not any of the cell functions typically. That's why you feel unwell, as well as on top of the actual immune response. Then we have assembly, which is exactly what it sounds like. Those things that you just made, the proteins and the genome, we're going to put them into actual functional virus particles. And then we're going to have them bud out or exocytose, or that means they're just basically the opposite of eating them up, pushing them out, like secreting, however that virus gets out. So that's typical for viruses that would be infecting a eukaryotic cell. If we're infecting a prokaryote, a bacterium, then that virus would be called, first of all, it would be called a bacteriophage. And second of all, they always burst out of the cell. They never bud out or anything. They also don't infect and go into the cell. They just land on the cell and inject their genome into it. And those pictures, you guys, if you look at the pictures of like bacteriophages, they look like they are robots or alien spaceships or something. That's actually what they look like. That's really what they look like. It's just, I feel like the most nuts looking thing there has ever existed. Okay, so that's our steps of infection. The cytopathic effect is just negative effects that would happen to the cell due to infection. So we're turning all of our attention back to making only viral particles and their parts. So that's going to have a negative impact on our cells. We're studying our entire outside of our cell with viral proteins. What were those viral proteins made for again? Those are the spikes. Those are the spikes that had the key to get into and dock with the next host cell. So if you're popping out spikes on the outside of the cell that we're going to bud out of and then coat the virus with, that's the goal. But it causes the cells to fuse with each other, to dock with each other sometimes. And that can, for example, cause syncytia, which is whenever the cells fuse together. And then you have this big old mass with all these different, it's basically multi-cell, but all fused together without membranes in between and nuclei and everything. So that's one possible thing. That's a cytopathic effect that you could see, as well as poor attachment of cells to whatever the extracellular matrix or whatever. Viral latency and what role does the provirus play? A provirus is when you take a viral genome and you stick it into the genome of the host. So our DNA, whenever we put a HIV into whatever cell it infected, that is the provirus whenever it's in the genome like that. And that does happen. That's how that works in that case. It also works that way with herpes viruses, very common. There's a lot of herpes viruses out there. Some people don't even think of them as being herpes virus. Things like Epstein-Barr that causes mono or chickenpox. Varicella zoster is a herpes virus. And surprise. So yeah, so that's the idea with the provirus. So it sticks its little genome into our genome, and then it can stay there, and it can wait for favorable conditions to go ahead and start reproducing again and hide out in that cell. That's viral latency. So whenever their genome is in our genome and hiding out, waiting for other conditions. So transformation is whenever we have taken a provirus and stuck it into a place that has caused our cells to not be able to function properly. So being infected with a virus like that, one, that kind of transforms our cells, literally, makes it so that our cells kind of will grow out of control. And that is an oncovirus. An oncovirus is any sort of virus that causes cellular transformation, growing out of control, leading to cancer. And this would include like HPV, right? Human papillomavirus, HPV, cervical cancer. We already mentioned about bacteriophages are viruses that infect bacteria. Lysogeny versus lytic. This is going back to that idea of latency, but now we're talking about in bacteria. So in bacteria, whenever they get infected with a virus, and that virus just replicates and then bursts out new viruses, that's the lytic cycle. However, temperate phages, which is a type of phage, these temperate phages are capable of going through a lysogenic phase. And the lysogenic phase is where they put their genome and insert it into the bacterial genome to hide out over time. And as bacteria divide, they're also copying that hidden genome from the prophage is what we call it, right? All right, so that's the genome from the temperate phage that's hiding out during lysogeny. So lysogeny or lysogenized bacteria, a lot of times whenever a bacterium, a pathogen, let's focus on those specifically, a pathogen is infected with its associated phage and undergoes lysogeny, incorporates the DNA from the phage into itself. That can allow the cell that has been infected to produce certain toxins that are associated with having been lysogenized by that phage. So genes kind of carried by the phage. This is pretty common in a lot of communicable pathogenic diseases, things like diphtheria and things like diphtheria is a good one, but Streptococcus pyogenes. Streptococcus pyogenes, man, it causes all sorts of diseases. We know it causes strep throat, but it also causes scarlet fever. And that rash that's associated with scarlet fever is caused by erythrogenic toxin. And that is a toxin that is made by the bacteria that have been lysogenized by a phage, this particular phage. That's pretty interesting, I think. So what is a prion? Or prion, however you want to pronounce it, I don't care, as long as you can see the word and write it or type it. And what kind of disease does it cause? Prions are essentially infectious, proteinaceous particles. They're protein, they're just a protein. And it's essentially a misfolded protein. We take a normal protein with a normal function and it's folded up wrong. It's not the right shape anymore. And for some reason, it seems like these wrong shaped misfolded proteins can come into contact with the right ones and cause those right ones to turn into the wrong ones. And then these can kind of like stick together or agglutinate together and cause holes in the brain that has an effect on the brain. So the diseases associated with these are called spongiform encephalopathies. So it's like mad cow disease. All right, that is chapter 6. Let's move on to 8, metabolism, one of my favorite things in the world. So metabolism, catabolism, anabolism. This should be pretty straightforward at this point. So catabolism, like cats, we're talking about breaking or tearing things apart. So breaking bonds, and when you break bonds, you release energy. And if we are releasing energy, we can store it in another form to be used later. That's the idea with catabolism. Breaking bonds, breaking down molecules, like when you eat, you're going to break the thing down and then get energy from that. Whereas anabolism is going to be the opposite of that. If we are going to be building up bigger molecules like new muscles or whatever, or however you want to look at it, new cells overall, right? If you're going to be building up these things, then you're going to need energy to be put into it. So anabolism is making new molecules, making bigger things, building new parts or new cells, and we need energy for that. So that usually is driven by energy from the catabolic processes. And all of the stuff together, all of the chemical processes going on within the cell, all together, the breaking and the making of bonds, that's metabolism. So that gets us into concept of what metabolism is. And then we have to understand what enzymes are. We've been talking about an enzyme for like a while now in a lot of these chapters. So I think most of us have an idea of what they are, but they're basically proteins that are catalysts. And a catalyst is something that helps a reaction to occur, essentially. We overcome activation energy, which is the energy that has to be put in. So we put in less energy now because the enzyme is there to help. Enzymes are, like I said, proteins that are going to allow you to do this. But they act on substrates. So whatever thing that they're going to start with, that starting thing, that's going to be your substrate. And then apoenzymes versus a cofactor coenzymes, holoenzymes, and all of these things. Holoenzyme is the entire functioning enzyme, all put together, because some of these proteins, in order to be functional enzymes, have to have things added on with them. Something like a cofactor, which could be like a metallic ion, or a coenzyme, which will be an organic molecule, an organic non-protein molecule. That will be like vitamins and stuff like that, often help out with the function of our enzymes. So the apoenzyme is that protein enzyme portion that will be actually doing the work. Then we have the adding in of the cofactor or the coenzyme that will help it actually do the work. You can't do it without it. And that whole thing that functions together is the whole enzyme. Catalytic sites or an active site is wherever we're going to be interacting with the substrate itself. The 6 major classes of enzymes, I'm not going to go into too much detail with these. We have the oxidoreductases, which are involved in electron exchange reactions. The transferases, which are going to remove a group from one thing and put it onto another thing. This often is related to substrate level phosphorylation, which is something important for energy production as well. Then we have isomerases, which has to do with changing structure of molecules. Liases, which is forming like bonds, like double bonds, and then ligases. Ligases is just like we had DNA ligase. We're reforming bonds that like phosphodiester bonds and stuff like that. And we're going to be putting the bonds back together, like from broken structures. And then we have hydrolysis, which is where we hydrolyze a molecule involving water, breaking down molecules. So yeah, so that's the idea of those enzymes, and that's all 6 of those. So the differences between exo enzymes and endoenzyme. So exo enzymes are enzymes that are going to be made within the cell and then secreted or pushed outside of the cell and function out there. An example would be when we talk about those fungi and them breaking down all these dead things, the saprobe fungi, right? Or the bacteria that do the same thing. They'll make their enzymes in their cell and then push it out to break down all the stuff from the dead matter, and then they can absorb the smaller broken down bits a lot easier than they could the bigger ones. So that's an example of an exoenzyme. Another exoenzyme might be one that allows a bacterium to break down penicillin. So that would be having to deal with drug resistance. An endoenzyme is just enzymes that'll be functioning within the actual cell. What are virulence factors or toxins? Toxins are going to be like exoenzymes that cause us harm. So that can include penicillinase, that enzyme that breaks down penicillin, because that's going to result in that bacteria being more pathogenic to us. That would be considered a toxin. They are exoenzymes that have negative effects on us whenever we're talking about a pathogen or an infection. But virulence factors, again, can't say it enough. These virulence factors are anything that helps a pathogen establish infection and create pathogenicity, which are negative effects from the infection. Denaturation is ruining the functional structure of a protein or a molecule of any kind. It could be nucleic acid as well. So heat is a good way to denature something, breaking up bonds, often breaking hydrogen bonds, which are weak bonds to begin with, or pH change, which also, if we're talking about pH, a lower pH being acidic, which means more hydrogen ions. So that could easily interfere with hydrogen bonds, right? That could definitely disrupt the whole relationship going on there with the hydrogen atoms in the structure, as well as in the actual fluid or milieu around it, body. So that completely changes the shape of the protein. And remember that protein functions are extremely closely related to their shape, just like we were talking about with the prions. We're changed the shape of that prion, of that protein, and now it's that infectious protein, the bad one. It can't do its normal function and it's causing damage in the cells. It's that simple. We can even have, when we talk about the effects of mutations in genomes, you can have one nucleotide change, just one A or T or C or G. Just one can be mutated, cause a different amino acid, because that different amino acid is in A, B, C, D place on the protein, and now it won't fold right, and that won't function at all, and now you've got a disease. It's that simple. So that's how serious this thing can get if you have misfolding of proteins. So one of the ways that we can control function protein, other than obvious one of denaturing it, we have competitive and non-competitive inhibition. In competitive inhibition, the inhibitor, the thing that's going to stop the process, the inhibitor binds in the actual active site where we would be acting on the substrate. It binds there, the substrate can't, the enzyme isn't doing its job to make, the resulting products, and the whole process gets shut down. In a non-competitive inhibition situation, now we are talking about a product usually that's made in the process. So we have enzyme A and we have substrate A, and they're coming together to do their work together to make product B. And that keeps going. Well, eventually there's enough product B. Product B can come and bind on a whole different part of that same protein enzyme A, that enzyme that's doing the work. And when it binds on that different part of that enzyme, causes a shape change due to the interacting of these molecules. And that shape change makes it so we can no longer bind to the substrate. That is basically telling that enzyme, you don't need to keep making the same, we've got enough of the product. So you can stop working now. So that is going to happen at a site. It's not going to be interacting at the active site. It's going to be acting at an area called the allosteric site or the regulatory site, same thing. Constitutive enzymes are just always being made. They can potentially be turned off and on based on these inhibition type things we just talked about. But a regulated enzyme is something that we will only be making as needed. So we won't actually be made from even the genetic level until we would need it. Yeah, it just depends on how often you need that enzyme and what you are using that enzyme for and how you might want to regulate it and everything like that. For catabolic and anabolic reactions, which ones are exergonic and which ones are endergonic, and what does that mean? Exergonic means giving off energy, releasing energy, and endergonic means it needs energy. We're putting in energy. If I were to do a chemical reaction and the cup got hot while I was doing it, that is releasing energy in the form of heat. That's exergonic. If I were doing a reaction and the cup got cold, actually cold, I didn't put ice in it, it just got cold from the reaction happening, that is an endergonic reaction, and it means it's kind of sucking heat out of and energy, therefore, out of the system. So catabolic reactions are going to be the ones releasing energy. So those are exergonic reactions, and anabolic reactions need energy, so they are endergonic reactions. The term oil rig, you can use oil rig, or you can say Leo says GER, that's what I do. But oil rig, that means oxidation is loss. So we're talking about loss of electrons. If a molecule lost an electron, then it's oxidized. If it gained an electron, then it is reduced. So rig means reduced is gain. Talking about electrons there. So those are redox reactions. Referring to redox reactions, those are going to be any exchange of electrons that would be happening within our reactions. And those are going to be carried out by oxidoreductases. Those are the enzymes that are involved in that sort of thing. The two major electron carriers in aerobic respiration, that's going to be NAD, which becomes NADH, and FAD, which becomes FADH2. So once they start carrying that H, they have an electron or two electrons associated with them. That's what that H represents on there. And that's important to remember for when we're talking about how aerobic respiration works. The major components of ATP, adenosine triphosphate. So this is going to consist of the nitrogenous base adenine, ribose, 5-carbon sugar ribose, and 3 phosphate groups. How does it supply the cell with energy? Well, those phosphate groups are highly negative and they're pushing away from one another, like 2 north on the magnets, like 2 north on knife. If you took 2 magnets and put the norths at each other, they're going to push each other away. Well, that's like what's going on with these phosphate groups on ATP. We've got 3 phosphate groups. They're pushing away from each other. They're constantly pushing each other away, but they're bonded together with covalent bonds, which remember they are sharing electrons. So that's what ATP is, and that's where it stores that energy, all that energy being stored from those phosphates pushing each other away. Aerobic respiration, anaerobic respiration, and fermentation. Just real quick, all of them involve glycolysis, okay? So all of them involve glycolysis. With fermentation, it's pretty much just glycolysis, and then at the end of glycolysis, you make pyruvic acid. That's just the resulting product of glycolysis. Always. So we have our pyruvic acid, and then in fermentation, they're going to turn that pyruvic acid into some sort of acid like lactic acid, or an alcohol like ethanol, like we drink. And that's how it deals with the NAD to NADH problem created in glycolysis. And it just does that over and over again. Glycolysis makes 4 ATPs, but it uses 2 ATPs. So you only get 2 as a product of it. So it's not a super efficient way of making energy, but it is a way for things like facultative anaerobes to make energy in the absence of oxygen. So if there's oxygen, they'll do aerobic respiration. If there's no oxygen, they'll do fermentation. So those are those guys. Anaerobic respiration. Okay, both aerobic and anaerobic. We've got glycolysis. Often you may have same steps going on in the Krebs cycle. That just depends on which organism we're talking about at that point. Then now we know in aerobic respiration, we have the electron transport chain where our final electron acceptor is oxygen. Well, anaerobes have essentially the same process, but they are going to use something else other than oxygen as their final electron acceptor. So just anything, whatever else. So that's what they're using. So that's the difference between those 3. The purpose of glycolysis is essentially to get our 6-carbon sugar of glucose and break it apart into 2 3-carbon pyruvic acids. That's the goal with glycolysis. We made a little bit of energy from it, but that wasn't even the point of the whole thing. The point of the whole thing is to get pyruvic acid. Then we're going to take our pyruvic acid and we're going to remove a carbon dioxide. This is the part where we get the carbon dioxide that you exhale. And then we're going to turn it into acetyl-CoA, whatever. Acetyl-CoA is what is going to go into the Krebs cycle. And we are going to remove another carbon dioxide in there through processing all this starting acetyl-CoA molecule into all these other structures like, citric acid or whatever. I don't need you to know the steps of it, but I do need you to know that we're going to be giving off carbon dioxide, and we are going to make NADH and FADH2 and some ATP. So all those 4 things. The good things that we want to use: ATP, NADH, FADH2, and then also carbon dioxide, because that's also made. So that's the Krebs cycle. What's the point of the Krebs cycle? The point of the Krebs cycle is to make NADH and FADH2. So cool. So we're going to put our NADH and our FADH2, and we're going to take those over to the electron transport chain or electron transport system. Then we are going to take our electron off of those NADH or FADH2. The electrons themselves are going to get shuttled through the cytochromes. So there's just a chain of some molecules. But as electrons get moved through these molecules and associate with these molecules in their little negative charge because they're electrons, that's going to cause hydrogen ions, which we have hydrogen ions now. When you take an electron out from the hydrogen, then you have hydrogen ions, H+, right? All these hydrogen ions, protons, if you want to call them that, get pumped out of the cell while a negative electron moves through. So negative electron moving through everything, pumping out all these positive charged hydrogen ions outside of the cell. So we created this proton gradient as we move this electron through. This proton gradient we call the proton motive force because it's about to motivate us to make some ATP. So get all these positives outside. We learn about diffusion and things like that and how things want to be equal on both sides of the membrane, essentially. So we want to equal out the hydrogen, the pluses on the outside with the inside. So they want to come back inside. The only way they can come back inside is through an enzyme called ATP synthase. And when protons move through, those positive charged protons move through the ATP synthase, it causes the ATP synthase to be able to perform the reaction of putting a new phosphate group onto a DP to make it a TP. And that's how ATP, our energy molecules, are made. So that's nice. So that whole process, that is called oxidative phosphorylation, because we have moving of electrons that led to the phosphorylation, adding on a phosphate group to create ATP. So there's that. So that's nice. So we had the movement of all these electrons through, but they don't just like, go off into the ether. Where did the electron go when it got to the end? And what about all these protons you're pumping back in? Well, what are these guys doing now? They just live there forever? No, of course not. Because what we're going to do is take some of these protons, take two of those protons, actually, the H plus and the H plus. We'll take two of those guys. We'll take two of those electrons. So now we can turn that H plus and H plus into H and H because they got their electrons. And we're going to associate those guys in order to make it a stable, usable molecule with half of an oxygen molecule. So that's going to take your H and your oxygen to make H2O as a byproduct. That's our byproduct of aerobic respiration. That's why we need oxygen as our final electron acceptor. That's why whenever I suffocate you with a pillow, you die because you cannot make ATP, because you cannot deal with those electrons and those hydrogens building up, and you cannot make any more ATP, so you die. You die without it. That's how quickly your body will use up all your ATP and you die. So that's why you breathe in oxygen right there. That's why your red blood cells carry you in some oxygen. And we already talked about why you breathe out carbon dioxide. The carbon dioxide is coming from breaking down essentially that glucose molecule through glycolysis and the Krebs cycle. And that's given off as our byproduct as we're stealing the electrons in the process to use in the electron transport chain. So that's how all of that works. So the carbon dioxide, a lot of people mistakenly think that when you breathe in oxygen and you exhale carbon dioxide, that oxygen from the actual air that you breathed in is the oxygen from into the carbon dioxide that you're breathing out. It's not the same, completely different oxygen. The oxygen from your carbon dioxide molecule came from glucose. Your oxygen that you're breathing in now is going into water, and you're going to pee that out later on. So that's what you get. So that's how that's aerobic respiration. This is why it's my favorite. How is that not the coolest thing? Anaerobes just use something other than oxygen in there. So they're going to make a different product as well. The net yield of ATP for prokaryotes and eukaryotes. Prokaryotes, they make 38 ATP through all of this net. Eukaryotes are going to make 36 because we have to deal with the membranes within the mitochondrion. So again, with eukaryotes like you and I, all of this is going on within the mitochondrion, which is an organelle in our cells. Bacteria do not have organelles, so don't forget that. So all of this is going to be happening in that cytoplasmic membrane, that inner membrane of the bacteria. This is happening on that instead of on the mitochondrial membrane. So what do we get from fermentation? We kind of already talked about that. Lactic acid, which can be useful for us as far as like making things like yogurt or trying to produce cheese products. Or we can have alcohol as a byproduct. Of course, we want that for drinking, for good old times. Also, a lot of these can create gases that are produced to allow us to have bread in it and everything like that. So that's kind of neat too. So that's fermentation, and so we are thankful for those facultative anaerobes. How does photosynthesis tie in? We mentioned this a little bit earlier, that photosynthesis works similarly to the electron transport chain. But instead of using electrons that we've put in from all these breaking down process, we are using light to excite molecules. And then when that's happening, we are allowed to pump those hydrogen ions out of a membrane again. And then when they come back in again through ATP synthase, same thing, we're going to make ATP. It doesn't make a lot. If it only make 2 ATP, and then we can use that 2 ATP in the dark reactions, which is totally independent from the light, in order to fix carbon dioxide into an organic molecule like glucose, which is just going to get shuttled right in to the nearby mitochondria of the cell if you're a eukaryote. And then that's going to be used to make energy for that cell, like a lot more energy. So that's the idea of how that is going to work. So that is everything for the metabolism. I'm going pretty quick on this. Do you have any questions? Okay, I know it's probably not that useful right now anyways, but it'll be good for rewatching later. Okay, and then our last one that we're going over is the chapter 9, which is the microbial genetics. A genome is all of the genetic information within that organism. So that can include the chromosome as well as any extra chromosomal or outside of the chromosome genetic information as well. So in bacteria, that often we are referring to plasmids. Eukaryotic cells, by the way, they can also have plasmids. These are little circular extra bits of DNA that are not essential for survival, but can help aid in surviving better, essentially. Things like drug resistance. That's an example of what they might make. The differences between eukaryotic and bacterial chromosomes. Eukaryotes tend to have linear and large and many chromosomes. The bacteria have circular chromosomes. So eukaryotes, linear, several. Bacteria have circular, usually one, can have more than one. It's unusual though. The genotype and the phenotype. This is what genes you're carrying versus what is being expressed. You might carry in the back of your genes somewhere a gene for some sort of mutation that you're not expressing because you've also got a good gene that works well, that isn't mutated. And you might not know that you carry it, or that you got it from your mom, or that you got it from your dad, or whatever. Or maybe, your dad had blonde hair and your mom had brunette hair and you're a brunette. Well, you're probably carrying the gene for blonde. It's just the brunette is going to supersede that. That's just examples, right? So those are your genes, whereas the phenotype is what you're actually seeing. So the brown hair is an example there. So that's what you actually see expressed. The two classes of nitrogenous bases, that's the pyrimidines and the purines. I'm not really going to go over the details of which ones are purines and pyrimidines, but just be aware of that they exist. Chargaff's rule, that's just the rule that says A binds with T and G binds with C. So you should probably know A binds with T and G binds with C. If I give you a genetic sequence that says, A, what is the complement to this genetically? What would bind with it? Then you would have to answer T. So know Chargaff's rule on which ones bind with which and be able to give me complementary sequences and understand it. Anti-parallel means that when we have, I don't have my tablet here. Is this? Yes. I'm just going to draw a couple lines here. Bear with me. I wish I had my tablet. Okay, so we know DNA is double-stranded. So, but one of them starts at this end and goes one way, and the other one starts at the other end and goes the opposite way. So that's referring to anti-parallel. What exactly is it talking to? What exactly is it talking about? The start we refer to as the 5' end, and it goes and towards the 3' end. And what does that mean? It just means we're talking about specific sugars on the 5-carbon sugar molecule for DNA. That's deoxyribose. There's a specific carbon atom on there that we have numbered all of them, and number 5 is at that end, and number 3 is open at the other end. That's all it means. It's just how those molecules are linked together. But 5' is the start and 3' is the finish. So one of them goes that way and the other one has to go the opposite way. So this one has to be 5' and 3'. And then we have our little ladder going on in the middle. Whatever, those are where our hydrogen bonds are bonding together, the A to the T and the G to the C, and according to Chargaff's rule. So that's the structure of the actual DNA itself, and of course it's in a helix wound up. Semi-conservative is referring to the fact that if I take 2 parent strands of DNA and I separate them apart and I make copies according to each of those, we would use one as a template and the other as a template. And so make copies based on A to T and G to C. Then our new ones that we've just made, our 2 double-stranded ones that we've just made, half of that is the old version and half is the new version. Half is the old template version and half is the new version for each of those. So that's semi-conservative. So that's all that's talking about. We have template version, original, and then the new one that we made the copy from. So every time that you copy DNA, that's going to be your result. Primers are needed during DNA replication because DNA polymerase does not add on nucleotides to nothing. It has to have something to add it on to. So we use typically RNA molecules as our primers, and then we'll go back in and fill them in with DNA later. So that's necessary during DNA replication, as well as PCR, polymerase chain reaction, which is where we've hijacked this system to use it in science. All right, so the leading and the lagging strand. Oh man, this goes back to that 5' to 3' thing. Basically, we separate out so DNA isn't that short, right? It's this big old long chain, big old. We're opening it up into this area, this loop area. That's our origin of replication, where we're opening up and we're starting out to start copying everything. DNA polymerase only adds nucleotides in the 5' to 3' direction. So once we start going and we're moving from 5' to 3', it's fine. We'll just keep going and pushing into the opening and opening and opening and opening of that origin replication and keep going. The other strand, however, remember, it's oriented the opposite way. So we have to add 5' to 3' to that strand too, but we can't because it's only presenting that starting edge as we're opening up. So what do? Do you wait till you open up the whole darn thing and then copy the whole thing in one go? No, what we do is as we open it, we make a fragment that is in the 5' to 3' direction of what space has been opened. And that fragment is called an Okazaki fragment. Okazaki fragments also need primers, just FYI. So we'll make that in fragments. And so as it opens up more and more of the space is open up. Now we can go back 5' to 3', make another fragment, open it up more. There's no more space now, open up. So we'll go 5' to 3', make another fragment. And what happens is after we have done all this and replaced our primers with DNA, we can seal all of those fragments together using DNA ligase, which is going to seal anytime you have DNA fragments and seal them up. We use DNA ligase. It's going to seal those phosphodiester bonds between the nucleotides. So that's essentially what's going to happen. So that strand obviously has a lot more going on with it than the other one, which is just going straight on down. The straight on down one we call the leading strand, the one that has to have a little bit of help that comes in fragments, the Okazaki fragments. That one is called the lagging strand. So that's what those terms are referring to. Transcription and translation. I mean, all right, so transcription, that's going to be taking DNA, our blueprint that is at home in the Smithsonian Institution. We go in there and we want to know how to make, I don't know, a sword. So we go into the Smithsonian Institution and we look specifically for the page about making a sword. And then we copy that page and then we take it home with us to go make the sword, right? The blueprint that we keep at the Smithsonian, that's your DNA, that's your genome. The copy that you made of the sword, how to make sword, that you took with you to go home, that's your messenger RNA. And then we're going to use that to make the sword. The sword is now the actual protein that's being made. So Smithsonian blueprint to the copy of how to make a sword, specifically from that whole big old thing. That's DNA to RNA. So that's transcription. And so that we did writing there, right? So we had to write a thing in that analogy. So transcribe, like scribes do, they write. So we've taken that home and now we're going to translate that whole wordage into a sword by building a sword with our blacksmithing skills, because apparently we do that. So that's essentially what's going on with transcription and translation. Transcription, DNA to RNA, translation is that messenger RNA into protein. The product of transcription, again, that's going to be the messenger RNA or RNA in general, basically. And then translation, the product is going to be that protein. So the machine that's going to drive translation, that's referring to the ribosome. So it's two parts that are going to come together and basically latch on to that messenger RNA. And then transfer RNAs are going to come in and interact with the message of the messenger RNA. And as they match up, they'll say, okay, this anticodon on the transfer RNA matches up with the codon of the messenger RNA. And based on that, you need to put here phenylalanine. You need to put here tryptophan. You need to put here glutamate, whatever amino acid, right? And then we link all those together with peptide bonds. That's all happening thanks to the ribosome. The ribosome is where all of this is going to be happening. That's the structure of mostly ribosomal RNA with a little bit of protein. That is the machine that's driving translation, essentially. So what type of RNA is the message? That's the messenger RNA, pretty standard there. What type of RNA is has the anticodon? That's going to be the transfer RNA. It's going to be transferring those amino acids. So if your codon, which is a 3 nucleotide sequence on the actual messenger RNA, each 3 nucleotides stands for 1 amino acid. So if your codon read, I don't know, AUA, because in RNA we use uracil, by the way. So if we, instead of thymine, we use uracil in RNA. So AUA, now your anticodon is going to read U because that's the matchup to it. And if those two match up, okay, this one is attached to alanine. And so that means we attach this alanine to what we've been adding on to. So that's how all of that works, how codons and anticodons and transfer RNAs are going to be happening. What type of amino acid does a stop codon code for? There are codons, those 3-letter nucleotides on the messenger, that do not code for any amino acid. They code for nothing. And those are called stop codons. There are no amino acid associated with those. They do cause the ribosome and the ribosome to dissociate and the whole thing to stop. So that's why they're called stop codons. They're also called nonsense codons because they're nonsense. They don't count for an amino acid. They count for nothing. They just cause the whole thing to fall apart, essentially. Nonsense. Nonsense often does that. All right, introns are a problem for prokaryotes, and why do we care about that? An intron is a part of our own genome in eukaryotic genomes that doesn't count for a thing. It doesn't code for anything. In fact, when we go from Smithsonian to make that copy of how to make a sword paper on our messenger RNA, that's basically like we're copying information that we didn't need. And so we'll just like black out or redact the parts that we don't want. So those are the introns. Introns don't want them. Exons want them. They code for a thing. So we will take exon to exon and splice those together by this thing called a spliceosome does that. Cuts out the crap we don't want, the introns, and links together the exons, the stuff that we do want. And now we have a readable actual messenger RNA. Bacteria do not have that. Bacteria do not do that. They want nothing to do with that. So whenever we want to hijack them as producers of our own proteins for studying or for making medicine, for example, we have to be sure that we are giving them a code that does not have any introns. So that's why that's important, because they can't deal with that. So what we'll do is go make our messenger RNA, cut out the introns. So we have a functional messenger RNA, and then we will reverse transcribe, okay? There's an enzyme that does this for us. So it takes that usable actual messenger RNA with the true codable protein, takes that and turns it back into DNA that we can, which we call cloned DNA. We can put that into the bacteria. No intron. Get rid of them. Inducible operons and repressible operons. Okay. So the lac operon is an inducible one. And if we're talking about the arginine one, that is the repressible one. So repressible ones are pretty much always on. The way that operons work is they essentially have a promoter region and an operator region. And that's where our repressor protein binds whenever it is doing its little work. And then there's a whole bunch of structural genes afterwards. In bacteria, they have a whole bunch of them strung together on one messenger RNA that all code for proteins that are involved in breaking down lactose in this case. So they can do that. We don't do that. We do one at a time. But this we're talking about operons are in bacteria only. So, so basically we have lactose come along. We eat this, but not very frequently. So the bacteria see this lactose, and basically the lactose itself will interact with the repressor that is already on the operator, cause it to change the shape, because that's what happens when proteins interact with other things. And it releases off the operator. Now RNA polymerase, which is going to make our messenger RNA, can go ahead and bind to its promoter where it always starts, and then move itself, little old self, on down and make the messenger RNAs that it needs to make for generating the proteins to break down lactose. We're not going to do that unless lactose is there. So if we run out of lactose, we stop eating it, it's not available anymore. Eventually, it'll disassociate from that repressor protein, and the repressor protein will bind back on and stop the whole thing all over again. So that's an inducible operator. We induced everything to work by adding in lactose. The repressible ones, like the arginine one that was discussed in the book, basically what they do is they're always on. We're always making this enzyme that helps us generate arginine, an amino acid. Okay, it helps the bacteria make it this amino acid. And that's all the genes that are associated in making that amino acid for the bacteria. Always making it. Well, let's say we have just a whole lot of arginine being made, and we just got a lot of it, and we're not using it because we're, I don't know, taking a day off, being lazy. We don't need it as much. So what happens is we get enough of that arginine product made, and it will go and it will bind with the repressor protein. Same concept here, right? Except this time it's going to change the shape and allow it to bind on to that operator region. So now we can't make any more. Polymerase can't work its thing. It's getting blocked by that repressor, and we'll make no more of that arginine. But whenever we need more and it runs out, the repressor protein dissociate, and we just go back to making more. So that's how a repressible operon works. That's how those two operons work. Horizontal gene transfer in bacteria. This is basically going to be talking about the sex pilus, which is conjugation. This is going to be talking about transformation, in which competent cells will be picking up DNA from their environment. So maybe from some dead cells, or maybe we made a plasmid that we're trying to put into them, for example. Code for some genes we want. And then the last one is transduction, which is allowing for DNA from a phage to be put into an infected bacterium by, like lysogeny and that sort of thing, which we talked about previously. So that's basically the three that are involved. Those are horizontal because they can be between bacteria that already exist, whereas vertical would be considered like if you got something from your parents, inherited genetically from your parents. So that's the difference between those terms there. So that's horizontal gene transfer. Major types of mutations. I really just want mostly to talk about, we have silent mutations where some of our codons are 3 nucleotide codons that code for a specific amino acid. Some of those we might have UAA and UAC, and they might both code for alanine. Well, so if you had a nucleotide change as a mutation from the UAA to UAC, well, you're still coding for alanine. Your protein doesn't know the difference. It's the same thing. So what does it care? It doesn't. That's a silent mutation. Still making the same amino acid. Until you change one amino acid, that is called a point mutation. Then we have the missense mutations. Those are going to be like if I had UAA to AAA. And so we mutated UAA to AAA. UAA does not code, well, UAA codes for alanine. AAA, I don't know, codes for phenylalanine. It's a whole different thing, amino acid. So no, we're not making the right amino acid now. That could have structural effects on that protein and functional effects on that protein as a result. So that is a change where we have changed one amino acid to another amino acid. That is called a missense mutation. However, let's say we went from UAA to UCA, I don't know, and UAA was our alanine, and now UCA doesn't code for an amino acid. UCA codes for no amino acid. It is nonsense. It is stop codon. So we've changed that one nucleotide and changed from our amino acid that we should have been making into a stop instead. It's a nonsense mutation. Nonsense. So there's no amino acid there. We also have frame shift mutations, which are pretty important to know about. A frame shift mutation is like if you had a single nucleotide deletion or addition, and it's going to shift basically the whole reading frame. So if it was AAA, AAA, and I insert in, into the third spot, C. So AAC, AAA, AAA, I don't know, whatever. That's going to change where you're reading what amino acid as you're going down the line for the sequence of that gene or the messenger RNA codons, right? So if completely change everything. So I even just want to write it down. I kind of write it on this screen. Let's try it. I don't know what that word means. Okay. Whatever. cares? Okay, so if we had A, faster, C, A, T, those would be U in the codons, but whatever. A, U, C. I'm real good with this. A, U, C. That's one codon, and then I don't know, C, C, G. And that's another codon, and I don't know, G, A, A. So that's our little sequence there. If I go in there and I insert into this point right here, a G, I don't know, I inserted one in there because of mutation, okay? So now we have A, U, G. Then we have C, C, C. And then we have G, G, A. And then A something. So you see now, if we read these codons, these are completely different amino acids potentially than CCG would give versus CCC potentially, right? That shifts the whole reading frame over. Same sort of thing happens if you had one nucleotide deletion. So that's a frame shift mutation that can be potentially devastating to protein production. You can also have mutations that take something that was mutated and turn it back into how it was originally. It's called a back mutation. So it's something that we can talk about here in just a second, actually. Yeah, the Ames test. Okay, that is where we are. All right, and so mutations, what happens if they're not repaired in time? Well, in time means like we're going through our checkpoints when we're dividing up our cells. If we don't catch our mutation at those points, they will become, you know, a fixture from then on. They're permanent and they can't be changed until they're carried forever. So that's a serious issue. So the Ames test, this is a pretty cool little test. Basically, they take some salmonella. This particular salmonella cannot make the amino acid histidine. You need it in order to build proteins. So it can't make histidine on its own. It would have to have it supplied if it wanted to live. So we're going to take those and we're going to plate them onto media that does not have histidine. So if you're doing that, they can't make the histidine and it's not being supplied, it shouldn't grow. So that's what the setup is. However, you take that same media that salmonella cannot make the histidine. We are not going to put it in the media, but we are going to put a mutagen in there or something that could potentially cause mutations in DNA. We want to know if it works or not. So we put that into the media and now suddenly our salmonella are growing. Why are they growing? Because they had a mutation that allows them now to make histidine on their own without it being supplied to them. So that's what is going on with the Ames test. If something, if they are given a chemical and that chemical allows that salmonella to make its own histidine when it shouldn't be able to, or grow on that media when it shouldn't be able to, that means that that chemical is likely a mutagen. And if it can cause mutations in bacteria, it can cause mutations in humans. And that's the Ames test and how we test that sort of stuff. So that is chapters 5, 6, 8, and 9. And do you have any questions? Okay. And so I'll see you guys for the next one, I guess. And have a good time.